So for the lecture today, we're going to be talking about specific catalytic mechanisms or strategies that enzymes use to catalyze their specific reactions. So just so we're on the same page, we'll start off with a quick review of some of the common features of enzymes that we've been talking about the last couple lectures, just so those have fully sunk in before we move on to more specific mechanisms. I'll do a quick outline of some common catalytic mechanisms that enzymes use, and then we'll spend the bulk of the lecture talking about one specific enzyme called chymotrypsin and look specifically how it catalyzes its particular chemical reaction. So this will actually be a pretty short lecture, but hopefully it'll solidify some of the topics we've discussed so far. So let's launch right into it. Lecture number 10, enzyme catalytic mechanisms. So before we launch into our discussion on enzyme catalytic mechanisms today, I wanted to go over a few features of enzymes that we've already covered, but that will come into play today, so I think it's important to go over them very quickly. So number one is that enzymes bind substrate at the active site. So they bind, you can see here's an enzyme, and it has this cleft right here, which is the active site, directly over the catalytic site, which is shown here in red. So when that happens, when enzymes bind substrate at the active site, it releases binding energy by that interaction between the substrate and the enzyme. And so the substrate specificity of each specific enzyme is going to be defined by the affinity for the active site residues with our particular substrate. So we talked about that when we talked about trypsin proteolysis versus thrombin proteolysis. So they have they catalyze the same reaction, which is the hydrolysis of a peptide bond, but trypsin has specificity for bonds that are directly after lysine or arginine residues, whereas thrombin hydrolyzes peptide bonds between an arginine and a glycine group. So that substrate specificity is going to be defined by the affinity for the active site with our particular substrate. And so we talked previously about catalytic efficiency, which is Kcat over Km. So Kcat is the turnover number, so how fast an enzyme converts substrate to product, and then Km describes the affinity of an enzyme for a particular substrate. So that catalytic efficiency is going to be optimized when an enzyme interacts with its ideal substrate. So these are substrates of chymotrypsin, and you can see that phenylalanine has the highest level of catalytic efficiency compared to other substrates. And then lastly, enzymes maximize their catalytic efficiency when they bind the substrate's transition state. So here, if you'll remember this figure, so we have a substrate that proceeds to product by first going through the transition state, and enzymes enzymes function to lower the energy of activation by binding that transition state, and that's what increases the rate of reaction. So these are just a few points before we get started today with specific catalytic mechanisms that I just want to emphasize when you're thinking about enzymes for the rest of the lecture. So there are a few different categories of common catalytic mechanisms that you'll encounter and that enzymes use to catalyze reactions. So the first one is covalent catalysis. So in covalent catalysis, the catalytic site of an enzyme will contain a reactive group. So this is usually a strong nucleophile. So a nucleophile is an electron-rich atom that's attracted to a positive charge on another group. And so this electron-rich atom, this nucleophile, will initiate a nucleophilic attack onto a substrate molecule. So the nucleophile will donate electrons and briefly become covalently modified during the reaction. So it'll actually form a covalent bond with the substrate at some point during the catalytic reaction. So that's covalent catalysis, and it's a very common catalytic mechanism. Another one is general acid-base catalysis. So this would be when a molecule other than water donates or accepts a proton during the course of a catalytic reaction. So amino acids in the active site are usually playing this role when we're talking about enzymes. And it's often histidine because histidine has a pKa that's near physiological pH of 7. So histidine is able to sort of more readily than other amino acids donate or accept a proton. So that's general acid-base catalysis, and it's also very common in enzymatic reactions. And the third category of common catalytic mechanisms is metal ion catalysis. So metal ions often act as cofactors for enzymes in catalysis. So a lot of different enzymes contain metal cofactors, and these are required for them performing their catalytic reactions. So metal ions can act as electrophiles that stabilize a negative charge in a reaction intermediate, or they can generate nucleophiles by increasing the acidity of nearby molecules. And by nearby molecules, we mean amino acid residues in the active site of a protein. Metal ions can also be involved in substrate binding, so they could increase the binding energy, which would increase the catalytic rate of the reaction. So that's metal ion catalysis, and that's also very common. It's very common for enzymes to have a metal ion as a cofactor and to use this in catalysis. And then the last category is catalysis by approximation and orientation. So for reactions where you have two distinct substrates, A and B, the enzyme facilitates the reaction by bringing them into close proximity. They also might need to be oriented in a specific way, and so the binding of these two substrates at the active site, just by bringing them in close together, that will increase the rate of the reaction between these two substrates. So the last one, catalysis by approximation and orientation. So now we can talk about principles of enzyme catalysis specifically, and to do this, we're going to use chymotrypsin as an example of an enzyme because it really uses a catalytic mechanism with a lot of different features that you'll come across in other enzymes across a lot of different classes. So chymotrypsin is a serine protease, just like we've talked about, just like with trypsin and thrombin, and it cleaves peptide bonds using a serine residue for catalysis. So the normal function of chymotrypsin is that it's secreted by the pancreas into the small intestine to aid digestion. So it's going to be breaking down peptide bonds from food that you eat to release amino acids from protein molecules that you ingest. So chymotrypsin preferentially cleaves on the carboxy side, so it cleaves that peptide bond on the carboxy side of hydrophobic aromatic amino acids. So that would be phenylalanine, tryptophan, and tyrosine. So those are the three hydrophobic aromatic amino acids. So chymotrypsin will cleave on the carboxy side of those amino acids, and it also has a lower activity against just other large hydrophobic amino acids like methionine and leucine. So how chymotrypsin does this, how it cleaves these peptide bonds, is that it uses what's called a catalytic triad in its active site. And this catalytic triad is made of residues serine 195, histidine 57, and aspartate 102. And so these three residues together will perform catalysis. If you mutate any one of these three residues, the enzyme will no longer be catalytically as active. So I want to emphasize that these residues are very distant in the primary sequence, but they're located close together when the protein is in its final folded form. So here is the actual primary sequence of chymotrypsin. You can see histidine 57 here, aspartate 102 here, and then serine 195 here. So they're far apart in the primary sequence, but when the protein folds into this globular shape here, you can see the active site is shown here in red. So if we look at the ribbon diagram and then zoom in, you can see when the protein folds, the serine 195, histidine 57, and aspartate 102 are located in very close proximity at the active site. And so these three residues make up the catalytic triad that chymotrypsin uses to actually catalyze its reaction. So these catalytic triad of residues in the chymotrypsin active site work together to catalyze the reaction. Like I said, you need all three of these to get the reaction to work. So serine 195, it functions by becoming a nucleophile that'll initiate a nucleophilic attack on the carbonyl group of that polypeptide chain. And we'll go into more detail here in a second on that. So what histidine 57 does, it removes a proton from serine 195, and that actually generates the alkoxide ion nucleophile of serine 195. So serine's not as strong of a nucleophile when it's protonated, but if it loses that proton and donates it to the nearby histidine molecule, then this is a negatively charged alkoxide ion and a very strong nucleophile that's going to be attracted to positively charged groups. So what aspartate 102 does, it's sort of just aiding this happening. So it orients histidine 57 to make it capable of accepting a proton from serine 195. So the ability for histidine to accept that proton from serine 195 is aided by this interaction right here from aspartate 102. So it'll both orient histidine and make a bond with this proton here, and that makes histidine a better electron acceptor. So these three residues all work together in the active site for chymotrypsin catalytic activity. So now that we've looked at the catalytic triad of residues involved in chymotrypsin, we can actually go through each of the steps of the catalytic reaction and see how these catalytic residues actually generate the reaction product. So we'll go through it step by step. So step one is just the substrate binding to the active site. So we have a substrate here, and so this shows the peptide bond that's going to be cleaved at this position right here, and then R1 is an aromatic amino acid, and then R2 would just be some other residue. So step one is that this peptide is actually bound at the active site, and that'll be through a bunch of interactions outside of the catalytic triad. Now step two is that serine and that alkoxide ion initiates a nucleophilic attack against the carbonyl carbon. So you can see right here, this alkoxide ion generates a nucleophilic attack on this carbonyl carbon, and then the oxygen or the electrons from that double bond will then go to the oxygen, and then what form is something called a tetrahedral intermediate. So now, since the oxygen is covalently bonded to the carbonyl carbon, this will form this tetrahedral intermediate, where the carbon is bound to, amino acid 1, and then there's the peptide bond there, and then it's also bound through a single bond to that oxygen, carbonyl oxygen, and then it's covalently attached to the serine residue on chymotrypsin. And this tetrahedral intermediate is going to be stabilized by what's called an oxyanion hole, which is just a feature of chymotrypsin. So it's stabilized by the backbone hydrogen bonds in this oxyanion hole. So this allows chymotrypsin to stabilize the reaction intermediate, or the transition state. In this case, it's called the tetrahedral intermediate. So we talked previously about how important that was. This is one of the mechanisms that chymotrypsin uses to stabilize that transition state or that tetrahedral intermediate, and it's through these hydrogen bonds from the amino groups in this oxyanion hole. So step 3 is actually going to be the collapse of that tetrahedral intermediate and the breaking of that peptide bond. And this is going to be facilitated by histidine acting as a general acid catalyst. So we can see here these electrons on the carbonyl oxygen that are in the tetrahedral intermediate are then going to form a double bond with the carbon again. And then the carbon, the peptide bond, the electrons from that are going to interact with the hydrogen atom that's now associated with histidine. And so this will make two different species, and one of which, the amine component, is going to be interacting with the proton on histidine, and that peptide bond, as you can see here, is going to be lost. And so that amine component is then going to be released, and that leaves an acyl enzyme intermediate with serine covalently attached to the carbonyl part of that peptide. And then step 5 is going to be the binding of water. So water is going to enter into the reaction. If you'll remember from our previous lecture, proteases like chymotrypsins are hydrolases that break chemical bonds by the addition of water. So this is where the water actually comes into this reaction. So the water is going to be interacting with histidine and between histidine and that acyl enzyme intermediate with the covalent bond between serine and the carbonyl component of the peptide chain. Then step 6 will be a nucleophilic attack now on the acyl enzyme intermediate. So histidine is going to act as a general base, and so it's going to accept a proton from water, and then the hydroxide ion is going to act as a nucleophile, which is going to attack the carbonyl group. So this actually forms another tetrahedral intermediate, and that's again going to be stabilized by the oxyanion hole. Now step 7 is going to be another collapse of that tetrahedral intermediate. And this is going to be facilitated by histidine, again, acting as a general acid catalyst. So it's going to donate a proton to serine. This covalent bond between serine and the peptide chain, the carbonyl component, is going to interact and be broken, and it's going to interact with the proton that's donated by histidine. And that's going to result in the collapse of this second tetrahedral intermediate in the reaction. And then the last step is going to be the release of the carboxy component of the peptide. So now we've broken the peptide bond and released both the amino and the carboxy component of the peptide. And that leaves the enzyme in its original state. So it's immediately ready to catalyze another reaction. So we just went through the catalytic mechanism of chymotrypsin, and I just want to point out that all serine proteases are going to use this same catalytic mechanism. So all serine proteases have identical catalytic triad residues. And really, all serine proteases have a similar overall tertiary structure. But we know that they have distinct substrate specificities. So chymotrypsin, of course, hydrolyzes after aromatic hydrophobic groups, and then trypsin hydrolyzes after positively charged lysine or arginine groups. So this distinct substrate specificity between these two enzymes is based on active site residues in an area known as the S1 pocket that's near the catalytic triad. So you can see here with chymotrypsin, it has a very deep pocket, and then it has a serine residue, serine 189, at the bottom of that pocket. So the catalytic triad is going to be adjacent to this region, like up in this region over here. But it's it has this deep S1 pocket and a serine 189 that allows it to interact with these bulky aromatic residues. Now, on the other hand, if you look at trypsin's S1 pocket, it's very different. So it has features the same. It's got these two glycine residues that make up a same sort of pocket, but the pocket is a bit more shallow because it has an aspartate 189, at the bottom of this S1 pocket. And that negative charge of aspartate 189 is going to be attracted to the positively charged lysine or arginine residues that are trypsin's preferred substrate. So something like a phenylalanine or a negatively charged amino acid, it's not going to be able to be accommodated in this S1 pocket. So the specificity of different enzymes, even those that use the same catalytic mechanism, is going to be dictated by residues that surround the active site and make interactions with a specific substrate. And so just lastly, also, since we're talking about catalytic mechanisms, I do want to mention that temperature and pH will have an effect on enzyme activity. So these are the two examples your book talks about. So increased temperature generally increases the reaction rate of all different types of enzymes. And this is due to Brownian motion, where just the molecule, the enzyme and then the molecule surrounding it are going to have a higher excitability in solution due to the increased Brownian motion at higher temperatures. And this increase is going to occur until basically the enzyme starts to become denatured, and then activity is obviously just going to drop off because it's no longer folded. Enzymes also usually have an optimal pH for activity based on those catalytic residues that actually perform the catalytic reaction. So you saw how important it was, particularly for like histidine activity in chymotrypsin, for it to be a general acid base donor and acceptor. And so there's going to be an optimal pH where that reaction is going to happen at its fastest rate. And so a serine protease like chymotrypsin is going to have a pH sort of curve like this, where the optimum activity is at about pH 8. But other proteases like pepsin, which is in a different class and will use a different catalytic residues, are going to have a different pH. And so pepsin is going to operate at a much lower pH than chymotrypsin. And this actually makes sense physiologically. So we know that chymotrypsin is in the small intestine. It's secreted by the pancreas, whereas pepsin is a protease that operates in the stomach, which is a very low pH environment. So it's been optimized through evolution to operate at a lower pH. And so this pH is going to have to do with the gaining the protonation and the deprotonation, usually of these different types of groups, which is outlined in this particular figure right here. All right, so I hope you all found the lecture interesting, and I hope you're really starting to get a good understanding of how enzymes work. So we reviewed a few features of enzyme catalysis, so binding energy, substrate specificity, catalytic efficiency, and the transition state binding. So those are clearly important topics to master. We then went through some of the different catalytic strategies that enzymes use, so covalent catalysis, acid-base catalysis, metal ions are used in catalysis, and catalysis by approximation and orientation. So for chymotrypsin, I don't expect you to remember every single step in its catalytic mechanism off the top of your head, but when you're studying it, please understand how its catalytic mechanism specifically relates to these concepts that we've been discussing. And it's also important to understand that the protein structure around the active site dictates that substrate specificity. So if you think about chymotrypsin in terms of trypsin and how their substrate specificity is different, even though they catalyze the same type of reaction, I hope that helps solidify that in your mind. And then also just remember that temperature and pH can affect enzyme activity. So thank you very much, and please let me know if you have any questions.